Hello, my name is Brett Gelfand. This is for Bio 200 Gut Microbiome Project. A gut microbiome is the types of proportions of microorganisms in a given environment. In this lab, we are looking inside the intestine or gut microbiome of a flat-haired retriever. In this experiment, we determine what the gut microbiome is for several animals. We do this by using many different kits, including the QIAGEN kit, which I'll discuss later, to help us purify the DNA. After analyzing the DNA, we can determine what diseases and diet the animal has. By analyzing the animal's gut microbiome, we can determine if the animal has diseases like Crohn's or IBS, both which affect digestion. Crohn's disease. Crohn's disease is a chronic disease that causes inflammation and irritation in the digestive tract. The gut microbiome is an easy way to determine if an animal has this disease. In the current study, the researchers found higher levels of amino acids and their derivatives in stool samples from the people with Crohn's disease. These were correlated with high levels of a group of gut bacteria called proteobacteria. IBS, or irritable bowel syndrome, is a common heterogeneous disorder characterized by abdominal pain associated with changes in bowel habits. Factors linked to IBS symptom development include history of enteric infection, modifications in the gut microbiota, altercations in brain-gut processing, and changes in visceral sensation and motility. We decided to analyze the flat-haired retriever's gut microbiome. We hypothesized that as the age of the animals change, so does the gut microbiome. The samples we compared came from 4 samples, coming from 2 different dogs, 2 different flat-haired retrievers. The fecal samples were collected 6 months apart from the same animals, just in a 6-month span. So we collected the sample one day, and 6 months later collected the next sample. This is so we could determine if age plays a factor in the gut microbiome of the animal. The first sample came from a dog named Hoshi, who was 1 and then 1.5 years old. And our second dog, Nova, ranged from 5 to 5.5 years old. Both of these dogs were female, weighed about the same weight, and were in very good health. 6 months later, the pets were not fed table scraps, yogurt, or these certain biscuits anymore. And they both switched their dry food to regular food. The only big difference was that Nova's exercise went from excessive to regular level. Materials and methods. The animals that compared for our gut microbiome projects were dogs, or flat-haired retrievers. We chose 4 dogs, but we ended up only being able to use 2 of them. As for their characteristics, they were from the same breed, flat-haired or flat-coated retriever. They all had the same weight, about 50 pounds, and were all females. The main difference was their age. The ages were 1.5, 5.5, 1, and 5. We then began our protocol. We ended up analyzing the gut microbiome. We used 3 steps for this. First, we isolated the DNA from the fecal sample using a commercial available kit. Next, a PCR reaction to amplify our specific region that we isolated. Next, we then quantified the DNA and tagged them with an index tag. The final step was determining the nucleotide sequence by using a next-gen sequencer. To isolate the DNA, we used a commercially available kit called the QIAGEN kit. This was used to purify DNA. Next, you put the stool sample in a buffer, and it prevents the enzyme from decreting the DNA. The DNA then binds to a filter and goes through a column to wash away any impurities. Purified DNA was then amplified 10 times, or tenfold, with the use of the Zymo kit. This makes the DNA concentrated. This was used similar to the QIAGEN kit, with the idea that you used buffer to wash away impurities. In order to check if this is right, we used an absorbance spectrophotometry. To determine the concentration of our DNA, we did this by using the nanodrop spectrophotometer. And we did not just check the concentration, we also checked the absorbance value. We did this so we could see the 260 over 280 value. We checked the concentration after, and we were able to find the volume from 100 milligrams of that sample. we then could do a proper dilution for the sample. So we ended up rinsing the impurities, we ended up increasing it tenfold so we could have more DNA to look at. We then ended up cleaning it all up and disinfecting it. We then performed amplicon PCR to amplify the regions of the 16S ribosomal RNA gene, specifically the V3 and V4 regions. Because it was a specific part in the ribosomal RNA gene particular to the bacteria region, with the V3 and V4 regions, those vary between different bacteria. So we can check with the database to see what percent of each bacteria is present. We then went to the gel electrophoresis. The gel separated DNA fragments of different sizes using an agarose gel to check if we amplified our DNA correctly. All steps were done in the way that the kit's manufacturer said it to be. So we end up going through all the steps that were labeled into the order of the instructions. It all came from the Zymo kit and the Kyogen kit, which was set up by the Kyogen Inc. or Zymo Inc. After checking our results, we noticed that our amplification did not work. We then decided to use group A sample for the rest. We then did PCR cleanup reactions. This cleans the components of the amplification buffer to get rid of the stuff in the way of the DNA, and the primers were removed as well. We then tagged the samples so we could tell the difference of what samples they came from. So when we sequence, we know what the samples come from. We then used a aromatic assay to determine the concentration of our sample, to make sure we had everything indexed and that we could actually sequence our results. We used the photometric assay because it is more accurate than the spectrophotometer. The DNA was then diluted to prepare for sequencing. Finally, we used the next-gen sequencer to determine our nucleotide sequence. We then showed our results. So from all the different samples, we have the different concentrations here, the different volumes, the volume of DNA water. This is the gel electrophoresis. As you can see, only half of them ended up working and the other half did not show any DNA concentration. That's why we ended up using group A's. And right below it is the gel electrophoresis and PCR cleanup. We then decided to use the order, and this shows at what level, at the order level, what bacteria were present. As you can see, Bacteroidealis was 71%, and all the other ones were a lot smaller. The genus had about 50% of it were Prevotella, and the rest were a lot smaller. And there were less than 3.5% of the total abundance was unclassified or defined as other. And unclassified, it also means could not be matched to any classification in the database. Then we had the discussion. Our experiment was successful in terms of quantity. To say there's a high enough concentration means DNA is pure. The absorbance ratio of a pure DNA must be in the range of 1.5 to 2.2. If it is below 1.5, then the DNA might have possible contamination. The contamination can come from misuse of the pipette, from us accidentally touching the sample, and many other ways. Because the product band was absent at 550 BP, we were able to see that the reaction did not actually work. That caused for us to end up using group A's sample. The presence of a PCR product band at 550 BP tells us that the reaction worked and there was no cross-contamination. The concentration for the DNA product was not adequate for DNA sequencing. We did not expect any products of PCR in the negative control. And so this is Bio 200. This is my Bio 200 product for gut microbiome.